For decades, the blood-brain barrier (BBB) has been regarded as the primary obstacle in the development of therapeutics for central nervous system (CNS) disorders. Consequently, most drug development efforts have focused on overcoming this barrier to achieve central efficacy. However, an emerging group of drugs are demonstrating that restricting BBB permeability can be a viable and advantageous strategy, particularly for therapies where the target site is peripheral and CNS side effects are a liability. This concept is exemplified by the first-in-class small molecule, peripherally restricted cannabinoid receptor agonist for the treatment of chronic pain. In this report, we explore the scientific rationale and translational strategy behind designing compounds that are excluded from the CNS. We will discuss how such an approach can enhance therapeutic precision, minimize adverse events, and accelerate commercialization pathways. If you would like to learn more about this innovative drug development strategy, please continue watching. Historically, the primary goal in developing small molecule therapeutics has been to maximize drug penetration into the brain to achieve central efficacy. However, this paradigm is increasingly being challenged for a growing class of therapeutic indications. In chronic pain, for example, where the target is the periphery and the central nervous system side effects are a liability, the goal becomes one of restricting BBB permeability. By limiting access to the brain, drugs can be designed to minimize the risk of neuropsychiatric and behavioral side effects, lower the potential for abuse and dependency, expand the therapeutic index, and ultimately improve patient compliance and safety. Moreover, excluding drugs from the CNS may reduce the regulatory burden associated with neurotoxicity studies and long-term CNS surveillance. Developing peripherally restricted compounds offers several advantages when transitioning from bench to bedside. First, it enables a more focused and efficient target product profile, as early discussions around brain penetration can help to align the TPP with the unmet medical need and payer expectations for a differentiated therapy. Second, avoiding BBB permeability allows for streamlined clinical development, as peripheral drugs may be eligible for accelerated development pathways due to their improved safety profile. Furthermore, endpoints can be focused on direct clinical outcomes without needing to evaluate CNS biomarkers or neurocognitive function. Third, a de-risked regulatory and market access strategy can be achieved through reduced CNS exposure, which may allow for a simplified safety data package and lower risk of regulatory delay. Notably, payers are increasingly favoring drugs with clearly defined safety benefits and mechanisms of action. Finally, manufacturing and distribution efficiency can be enhanced, as peripheral compounds typically do not require complex formulation strategies or invasive administration routes, which can impact cost of goods sold and ultimately a product's commercial success. Moreover, targeting cryptic or allosteric peripheral binding sites can provide robust intellectual property protection, and by differentiating from CNS penetrant competitors, brands can establish a unique position in the market and potentially extend their product's life cycle. In conclusion, the traditional view that maximizing BBB permeability is always the ultimate goal in drug development is rapidly evolving. For an increasingly large class of indications, particularly those with peripheral targets, designing drugs that intentionally avoid the CNS offers substantial scientific, clinical, and commercial advantages. A translational strategy focused on peripheral efficacy can yield safer, more targeted therapies and unlock faster, less risky commercialization paths. This represents a significant paradigm shift in drug development, one where precision is defined not only at the molecular level, but also at the pharmacokinetic level, including the blood-brain barrier.